As I mentioned, you have seen the classical SDS page and blue native page. The concept is similar and it can be applied in the two dimensional electrophoresis. There is little variation here, because now you do not need to add a comb. You have to have a flat comb for making a well to add your IPG strip, and you do not need to add the separation for stacking gel. So, in this laboratory demonstration video, I will show you for the two dimensional electrophoresis how to make the SDS page.
Protein separation by SDS page. SDS page, which constitutes the second dimension of two-dimensional gel electrophoresis, involves assembly of the gel apparatus, gel casting, equilibration of the IPG strip, followed by placement of the IPG strip on the gel and protein separation. Assembly of gel apparatus and gel casting. Clean all the components of the gel assembly thoroughly with water and dry them with paper towels. Carefully arrange the glass plates in the gel casting assembly, interspersed with separator sheets depending on the number of gels to be run. Assembly should be tightly packed such that there are no leaks. Close the casting assembly and tighten the screws provided. Prepare the gel casting solution consisting of acrylamide bisacrylamide, tris chloride, SDS, APS and timid, and pour it with the help of a funnel into the central channel of the casting assembly. APS acts as a free radical generator, while timid functions as a catalyst for the polymerization reaction. Ensure that the solution spreads evenly such that the top edge of the gel is uniform. Allow the solution to stand for polymerization to occur and spray a solution of SDS across the top to ensure that the gel does not become dry. SDS page. Drain out the excess liquid of the second equilibration solution from the IPG strip using a tissue paper. Soak the strip for few minutes in the same buffer solution that is to be used in the gel tank. Then carefully place the strip in between the glass plates and gently push it in until it rests firmly on the gel surface without any gaps. Place the filter paper containing the molecular weight marker beyond the positive end of the strip. Then pour the lukewarm agarose overlay solution over the strip to prevent the gel from drying up. Assemble the remaining plates in the gel assembly and carefully lower the entire apparatus into the gel tank containing the buffer solution. buffer level does not rise above the mark indicated. Place the separating chamber above this and pour the buffer solution into the upper chamber. Close the gel assembly and connect the apparatus to the electricity supply. Set the appropriate voltage and begin the run. All the proteins present on the IPG strip carry a large negative charge due to the action of SDS, which denatures the protein and binds to the polypeptide backbone at a constant weight ratio. This ensures that the proteins present get separated exclusively on the basis of their molecular weights rather than their mass to charge ratios as in a native page. The proteins having lower molecular weights have high mobility and migrate further through the gel pores, while the high molecular weight proteins remain closer to the point of application. This allows efficient separation of the proteins in the second dimension.
So, now we will talk about how to stain the gel and visualize the protein spot, because so far now you are able to separate the protein based on their pi, based on their molecular weight, and now everything is on a transparent gel. You do not know how well your separation has occurred. So, now you need to visualize your spots. So, different type of staining methods have been developed and those can be applied to visualize your protein bands or your protein spot, depending upon what type of gel you are using. So, after giving you an overview of staining techniques, let me describe you few staining methods in more detail. Let us start with Kumasi blue staining. This is one of the most commonly used stain for protein detection in polyacrylamide gels. The sensitivity is good, but not very sensitive as compared to the silver or cypro ruby, but its ease of performing the staining method, stability, the cost very less, and good compatibility with mass spectrometry. All of this makes Kumasi blue as a stain of choice for most of the laboratories worldwide. Now, there are some advanced form of Kumasi stains being developed, including biosafe Kumasi stain, which is non-hazardous, made keeping in mind the environmental scenario. So, it can be disposed as non-hazardous waste. The biosafe Kumasi is also ready to use stain and a single reagent protein stain. So, I will show you one animation how to perform the Kumasi blue staining. In this animation, I will show you how the staining can be performed by using Kumasi brilliant blue stain. The completed electrophoresis gel is placed in a tray containing the Kumasi blue staining solution, typically R250, that has been dissolved in an aqueous solution of methanol and acetic acid. The negatively charged Kumasi dye interacts with protein through ionic and non-covalent interactions. After adding the stain, the tray is then placed on a mechanical rocker, which allows for uniform contact of the gel with the solution by means of gentle rocking. After overnight stain, you have to drain out the staining solution. The stained gel is then placed in a destaining solution. Which consists of 50% methanol and 10% acetic acid to remove any excess dye that may be bound to the gel. Again, this process involves overnight steps or at least 10 to 12 hours of gentle shaking on the rocker. The stained gel can finally be viewed as shown in this image here, showing a 2D gel stained with Kumasi brilliant blue, and it can be scanned by using a scanner, and these protein spots can be easily viewed. So, in this animation you are able to see, after performing the 2D electrophoresis, how to add the Kumasi stain on your gel, do the staining and destaining procedure, so that you can visualize your blue spots on the transparent background. Each of the spot can be now used further for image analysis purpose. So, most of the time, if you are lucky and if your protein extraction protocol is good, you have good protein yield, but often your samples are very challenging. Despite your good extraction protocol and your good laboratory skill, still you are unable to obtain a very good protein quantity. So, how to move forward? You want to still do the separation of the protein, but Kumasi stain may not be sufficient to visualize the spot on the gel. Now, you need few more sensitive stains such as silver stain. So, I will describe you how to do silver staining. The silver staining is more sensitive than the Kumasi blue dye. First of all, when you have run your 2DE gel, you need to do the protein fixation in the gel by adding methanol and acetic acid. The staining procedure involves silver amine complex, which is bound to the tungstosilicic acid. The silver ions are transferred from the tungstosilicic acid to the proteins. Now, by providing alkaline solution with formaldehyde, it can reduce the silver ions to the metallic silver form, and the brownish tinge you can see on the spot, which can be further analyzed. But if you allow this reaction to occur continuously, you will see a very dark background on the silver stained gel. So, you need to stop the reaction by adding acetic acid. I will now show you this animation how to perform the silver staining procedure. In this animation of silver staining, I will describe you the procedure how to stain the gel by using silver stain. The completed gel is first placed in a fixing solution of methanol and acetic acid that fixes the protein bands in the gel and minimizes any diffusion. This step must be subjected to gentle shaking for around 30 minutes, after which the silver stain solution is added. After adding the silver solution, the gels are rocked gently in order to allow for proper and uniform staining. The silver stained gels are first washed to remove any excess stain and then placed in a developing solution where the silver ions get reduced to metallic silver. Formaldehyde in an alkaline solution in the presence of sodium carbonate or other alkaline buffers are commonly used for this process. Again, a gentle rocking will allow for uniform staining pattern. Finally, the gel can be viewed as dark bands or dark spots against a light background. So, let us start with cyproruby staining. This is one of the fluorescent stain which is easily visualized on the gel in the UV or blue light transluminescence. This stain is very sensitive and it is endpoint stain. So, very little background is used, unlike the silver stain where there is a lot of issues with the very dark background. The cyproruby has overcome those limitations. This is also one of the very sensitive stain and it can also be used for studying different type of post translational modification such as glycoproteins, often even low molecular weight proteins which are low abundance protein, those can also be visualized by using this type of stain. So, cyproruby is one of the very useful stain which is very compatible for doing further processing with the mass spectrometry. So, if you want to separate the proteins, you want to visualize them, after that your end aim is to characterize those proteins, what those proteins are, and further you want to do experiments on those. In that light, cyproruby is very compatible for mass spectrometry and you can further characterize your proteins. So, now I will show you the steps involved in performing cyproruby staining. So, I will show you one animation. Cyproruby red. The completed gel is first placed in a fixing solution of methanol and acetic acid that fixes the protein bands in the gel and minimizes any diffusion. This must be subjected to gentle shaking for around half an hour. After fixation step, the gel is soaked in the fluorescent cyproruby red stain solution. Gel should be kept on the mechanical rocker for uniform shaking. During the process, the gel gets uniform staining with the ruthenium based chelate dye. After this overnight step of staining, the gel can be washed with methanol and acetic acid solution. Again, you need to keep it on rocker so that uniform destaining can be performed. The MS compatible gel is then viewed using UV or laser scanning instrument with an excitation wavelength near 280 or 450 nanometers. This stain has two excitation maxima and an emission maxima of 610 nanometer. In this animation, you can see a gel pattern which is showing you the staining obtained after cyproruby red, and it is showing separation in 2 dimension based on the molecular weight and isoelectric point. So, now after looking at this animation, you are familiar that there are different type of staining methods can be used. Now, let me introduce you to another stain which is ProQ diamond, which is used for studying the phosphorylation of the post translationally modified proteins. Now, ProQ diamond, that is a dye, a fluorescent dye, which is capable of detecting phosphorylation. So, if your proteins having some phosphorylation, addition of certain phosphate residues, now this stain is quite sensitive to pick up those patterns of phosphorylation or post translational modification in your gel. This is very suitable for use in electrophoresis, and it offers sensitivity in nanogram level. Now, let us say you want to look at the post translational modified forms of the protein, but after that you would also like to know which proteins it belong to. You would like to do the mass spectrometry on these. So, often it becomes very difficult if you have just done the ProQ diamond on one gel. So, you would like to do the dual staining. So, first you would like to detect only your post translationally modified form of the protein or the phosphorylated forms, then followed by the same gel can be used for further staining with some endpoint stain such as cyproruby. Now, you take the same gel, now further stain with the cyproruby. So, you will obtain two images for the same gel, where few spots can show the PTM form and the overall the global pattern of the proteins can be visualized on different gel. So, in this regard, by comparing the PTM form or the phosphorylated form along with the complete profile of the gel, one can obtain the very comprehensive picture. This many proteins are separated on the gel, among those certain numbers are phosphorylated. One can then further excise those spots and do the mass spectrometry for its further identification and characterization. Now, let me show you this animation for doing ProQ diamond staining followed by dual staining. ProQ diamond, this is very useful staining technique for detection of phosphoproteins, and when used in combination with cyproruby red, it also provides a very comparative profile of total protein contents and the phosphoprotein content. The first step is to perform the fixation and followed by addition of ProQ diamond stain. The staining procedure is similar to the cyproruby red, which was earlier described. This fluorescent dye offers more sensitivity down to nanogram level and detects specifically the phosphorylation taking place at serine, threonine or tyrosine residues. For uniform staining, you need to add the staining solution for at least 8 hours or overnight. Now, staining solution can be removed and further washed with methanol and acetic acid. For uniform destaining, it is good idea to keep it on gentle shaking for at least 6 hours. Gel can be further scanned and image can be procured, especially at emission maxima of 580 nanometer, and it provides again on the 2 dimensions decreasing molecular weight and increasing pH information for all of these protein spots. Often people combine this phosphor stain along with cyproruby stain to obtain dual staining information, which was described in the lecture. So, in the animation you have seen that how ProQ diamond can be used to detect post translation modified form or the phosphorylated form of the proteins, and then other staining procedure such as cyproruby can be used for dual staining. Now, dual staining allows more than one detection protocol on the same gel. So, it has a lot of merit, and I hope after watching this animation you must be able to appreciate that. So, after discussing about different type of staining procedures which are available, now you have separated the protein spots on the gel. Now, you have visualized that by using different type of stains. Again, the selection of stain depends upon the type of application you are looking for. Often you have limitations of your protein. If you did not have enough protein, you probably used silver or cyproruby. If you had enough protein, you went with Kumasi brilliant blue staining. You were interested in looking certain form of PTM, you used ProQ diamond or other specific stains. Now, if you want to do some more advanced form to eliminate lot of gel artifacts, probably you used styrene dyes. So, after all of this type of staining detection, now you are ready to scan the image, because now you can see all the protein spots available on the gel in different colors, whether it is blue in Kumasi or brown in silver, pinkish in cypro, or different type of fluorescence pattern in styrene dyes and ProQ diamond. So, in a nutshell, here you can obtain information for various stains which are available, their properties and their sensitivity. Please note, the sensitivity is only approximate.
Gel staining and destaining. This process involves removal of the gel from the electrophoresis assembly, followed by treatment with a fixing solution, staining solution, and finally the destaining solution. Gel removal and protein fixation. Soak the gel present between the glass plates in fresh distilled water to remove any bound SDS. Pour the fixing solution containing methanol and acetic acid into another tray. Open the glass plates and carefully remove the IPG strip from the gel. Then, transfer the gel into the fixing solution by gently sliding it off from the glass plate. Place the tray on a mechanical shaker for an hour to ensure that the protein spots get fixed onto the gel, thereby minimizing any diffusion. Gel staining. Transfer the gel carefully into another tray containing the Kumasi blue staining solution and place it on the shaker to ensure uniform contact of the gel with the solution. The negatively charged Kumasi dye interacts with protein through ionic and other non-covalent interactions, thereby staining them with a blue color. Add some more staining solution such that the gel is completely immersed and leave it overnight for around 8 to 10 hours. Destaining. Drain out the staining solution from the tray and pour the destaining solution consisting of methanol and acetic acid into the tray. Place the tray on the shaker for around 6 to 8 hours until the background stain of the gel is completely removed and the spots are clearly visible.
So, we will continue in the next lecture. I hope you are able to appreciate the gel based proteomics and its potential. Thank you.